Hi. So, in this session we will discuss about the pattern recognition receptor, and we will take up in more detail about the toll-like receptor. In previous session we have discussed the origin of pattern recognition receptor concept, okay, and that was quite interesting. And after the discovery of pattern recognition receptor, the view of immunologist is completely changed. Okay, it's previously we thought that it's a non-specific thing and it's not so important, but now we know it is very important. Okay, there is some broad sensors, and these broad sensors eventually sense the microbial pathogen, and they play a pivotal role in development of adaptive immunity. Okay, so in this session I will talk about TLR. So, what is TLR? TLR is basically the toll-like receptor. The toll word came from, if you remember my previous session, you can understand that toll protein was discovered in the Drosophila, and protein similar to the toll, Drosophila toll, in human and in mammals was discovered, and we call it as a toll-like receptors. Okay. So, this toll-like receptors are basically recognizing all kinds of microbial pathogen, that includes bacteria, fungi, viruses, and the most interesting thing is that they recognize these entities on cell surface, as well as it also recognizes inside the vesicles inside, which is present inside the cells. Okay, we call it as an endosome, and it depends on the stage of this endosome. We can call it as a phagosome, phagolysosome, so and so. Okay, and upon recognition they activate, of course, the cascade of signaling, and eventually it results to the production of pro-inflammatory cytokines. Okay, and in addition to pro-inflammatory cytokine, they also induce a antiviral factor, or it's an antiviral cytokine, you can say that, and this we call it as a, broadly we call it as a type 1 interferon. Besides this, in recent past it was also reported that there is a type 3 interferons, which is also playing very important role in antiviral immunity. Okay. So, this is about the toll-like receptors, which is quite brief, and I will talk extensively about toll-like receptor in subsequent slide. But before going to that, I just want to introduce, there are some more pattern recognition receptor, or there is more families of pattern recognition receptor. This is much more appropriate to say that families of pattern recognition receptor. Another very important family of pattern recognition receptor is Rigi-like receptor. Here you can see this Rigi-like receptor is derived from Rig-I. This is a retinoic acid induced gene. Okay, and there is a family of molecule which is induced by retinoic acid. Okay, these sensors are very important against RNA viruses, because they sense the RNA molecule, and therefore they play a crucial role in sensing RNA from RNA viruses. In addition, they may also play an important role in sensing DNA virus. As you can see that when DNA virus replicates, then there is some RNA intermediate, and that RNA can be sensed, and then that will induce the response. And the responses are, again, this induces pro-inflammatory cytokine and type 1 and type 3 interferons, and they are playing very important role in antiviral immunity. Okay, this Rigi-like receptors. Okay, another member is the nod-like receptor. Okay, and this is also an intracellular sensor. Rigi is like Rigi is also intracellular sensor, okay. Nod-like receptors are also intracellular sensors, and they sense the signature from bacteria in the cytoplasm, okay, and then that will basically induce pro-inflammatory cytokines, and they play a very important role in production of one very potent family of cytokine, which we call it as a IL-1 family cytokine, and this is basically mediated by some multi-protein complex are formed in the cell upon sensing of some microbial PAMP. Okay, PAMP is basically pathogen associated molecular pattern. Okay, so upon sensing these sensors basically induce either inflammatory cytokine and or they will make some multi-protein complex, and then it will play important role in production of IL-1 family cytokine. Okay. There are some more sensors which plays a very important role in DNA sensing. Here I have not shown that those sensors. I will talk in more detail about the DNA sensors in subsequent sessions. Okay. So DNA sensors are also there, which senses the DNA molecule from pathogen, and then it induces the appropriate immune response. Okay. So let's move on to take a look how these sensors are located. Okay. So here you can see, as I have explained you in previous slide, this TLR4, TLR2, which is making a heterodimer with 6, and TLR1 also make a heterodimer with 2. They are located on the cell membrane, and TLR11 and TLR5 is also located over the cell membrane, and they sense their respective PAMPs. Okay. I will discuss about the PAMPs and their expression in subsequent slide. Okay. And here you can see that TLR7, 9 and 3 and 8, they are expressing in endosome. Here I did not show it. So here you can see that these sensors basically sense the PAMPs in endosome, and this is very interesting, right? Many viruses, if you see carefully, many of these viruses are basically endocytosed through this phagosome. Okay. So over there they get the protein or coat protein are dislodged, and then the nucleic acid come in picture. Okay. So this nucleic acid can be sensed, and this is very appropriately located. Okay. The sensors are very appropriately located, and they sense, and then this will induce the immune response. Another family of molecule is RLRs. Here you can see that members of RLR are located in cytoplasm, and some of this virus infection, they are basically come in the cytoplasm. Okay, the nucleic acid comes in the cytoplasm, and over there this can be sensed, and then there will be a appropriate immune response. Another is a nod-like receptor. So basically nod-like receptors, there are two major categories of nod-like receptor. One is nod 1 and nod 2, which one category is made of nod 1 and nod 2. They basically senses the bacterial signature molecule, such as some component of peptide or glycan, and there is another group of sensor which basically oligomerize and then they make some multi-protein complex, and then they will induce IL-1 family cytokine. So this we will study in great detail when we will take up the nod-like receptor. Here although I have given in a schematic, there is a production of IL-1 beta which is synthesized in inactive form, and this inactive form is basically converted into the active form by this multi-protein complex known as inflammasome. We will discuss in great detail in NLRs. Okay. Now let's come back to the toll-like receptors. So toll-like receptors, they have a 3 distinct region. If you see very carefully, the first region, which we call it as a leucine rich repeat. Okay, this leucine rich repeat is playing important role in sensing the PAMP. Okay, and basically these TLRs are consist of 18 to 25 copies of leucine rich repeat, and each leucine rich repeat is consist of about 20 to 25 amino acids. Okay, and they make a one unique structure which we call it as a horseshoe like structure, and then they sense the PAMP. In some cases, in some TLR, this leucine rich repeat is undergo cleavage in order to sense the PAMP. Okay, and generally these TLRs are basically expressed in the endosome. So all most of these nucleic acid sensors, they undergo cleavage, and over there will be a generation of active sensor which will sense the nucleic acid, and then it will transduce the signal. Okay, and this cleavage is basically mediated by various proteases like cathepsins. Okay, cathepsin family protein. Okay. The another region is the transmembrane region, which we also call it as a type 1 transmembrane region. Okay, and this is basically consist of mainly you can understand this is consist of a hydrophobic amino acid. Okay, and this plays a important role in inserting in the membrane. Okay, and the another most important region is the TIR region. Okay, here you can see that toll IL-1 receptor region, and this toll IL-1 receptor region is the cytoplasmic region, and this plays the most important role in signaling. So if you eliminate this domain, then there will be no signaling. And this name, the TIR name, came from IL-1 family receptor. Here you can see. So there are various IL-1 receptor subfamilies, and you can see that all these receptors, they have a TIR domain. Okay, this is basically IL-1 receptor domain. Okay, which is playing a very important role in downstream signaling upon binding with the respective ligand, that is IL-1 or IL-18. Okay, so this TIR domain is playing very important role in downstream signaling, and this TIR domain is also present in some of adapter molecule of TLR. As you can see, there is a MyD88. Okay, this MyD88 is also having this TIR domain, and there is another adapter molecule known as MAL or TRAP. So these proteins or these adapter proteins has this TIR domain. Okay. Now I will talk you about the PAMPs recognized by TLR. Okay. So there are if you see the TLRs, then you can find out that these TLRs can sense various biomolecule derived from the microbial pathogen. Okay, they can sense lipid or lipid derivatives, they can sense protein, they can sense nucleic acid. So these are the major biomolecule, okay, in living system. Okay, here you can see that the heterodimer of TLR 1, 2 and TLR 6 basically they sense triacyl lipopeptide and diacyl lipopeptide respectively, and TLR 4 which sense can sense the LPS. Okay, and this LPS is the sensing of LPS is not so simple, and you can understand in previous session I have talked about the work of Bruce Butler. So the TLR field basically discovered through the discovery of TLR 4, and sensing of LPS is little complicated. For sensing of LPS you need various molecule, as you can see in subsequent slide. You need a CD14, you need MD2, and there is one more protein known as LBP, LPS binding protein. So all these things are needed in order to induce the LPS or TLR4 mediated signaling. Okay. TLR can sense the protein. If you see carefully that TLR5 senses flagellin, and this flagellin is derived from flagellated bacteria. In addition, they can sense uropathogenic bacteria, and TLR3, 7, 8 and 9, they can sense nucleic acid. Like TLR3 basically senses double stranded RNA molecule, TLR7 and 8 senses single stranded RNA molecule, and TLR9 senses CpG motif DNA, which is hypomethylated. Okay. So here I just want to give one important message that that this pattern recognition receptor are germline encoded receptor, which is not the case in case of adaptive immunity. Okay. Over there, the receptors like B cell receptors and T cell receptors, they are generated after genetic rearrangement. Okay. There is a recombination is taking place. Okay, where the gene segments are joining and then they are making a receptor. Okay. Here in case of innate immune receptor, they are germ-line encoded receptors, and if you see very carefully, then you can see that all those molecules which are come in category of PAMPs, they are very important for these microbial pathogen. Okay, like peptidoglycan, LPS, nucleic acid, but they these sensors basically sense the distinguishing feature. Okay, like hypomethylated nucleic acid, okay, single stranded RNA, phosphorylated RNA, so on and so forth. Okay, and like a human cell, they don't have a peptidoglycan, so they sense the peptidoglycan. So here I just gave you the overview about the various PAMP, and in next slide I will show you the great detail of these PAMPs, what are the PAMPs and where these sensors express. Here you can see that TLR2 in association with 1 and TLR2 in association with 6, they sense lipoarabinomannan, lipoproteins, lipotecoic acid, cell wall from fungi, which is consist of beta glucan, and zymosin, which is derived from fungi, and they are expressing in variety of cells like monocyte, DCs, mast cells, eosinophil, basophil. So if you see this table very carefully, then you will notice that most of TLRs are expressed in macrophages and dendritic cells. Okay, except few. If you see TLR7, it is not expressing in DCs, rather it is expressing in PDCs. Okay, which basically produce ton amount of type 1 interferon upon viral infection. Okay, so you can go through this table and you try to understand and try to remember that these TLR are playing a very important role in various compartment of the cell. Okay, and here you can see that TLR10 is only expressing in human, whereas TLR11, 12 and 13 only expressing in mouse. Okay, so with this I will stop here, and in next session I will talk about the TLR signaling. Thank you.